I am waiting for. Yep, we're ready. Okay, so I am just want to make sure I mute so you don't hear my echo again because I know that happened last time. And welcome to the first installment of the Hot Zone book discussion that we're having. Um, and my goal today is to have a really great conversation with you that will hopefully tie the book and what you were reading and the whole study and outbreak of the Ebola virus and all the things that you saw in that book to what's kind of going on today and how scientists are able to actually study things like Ebola or coronavirus, which are, submicroscopic. They're extremely, extremely small, unless you saw in the book the hot zone where you had huge bricks of virus together um, that became problematic that you were able to see. Otherwise, viruses themselves are pretty much like submicroscopic. So, with that being said, I want to have a great conversation with you today. Take a minute, really quick, and just give me the hey on the comment bar to make sure that you are with me, and I know that we are ready to roll. Right now, I think Lily might be the only person. Lily, you're in for a really great conversation with me. Lily might be the only person who's here. So if anyone else is here, just please give me the thumbs up and let me know that we're ready to go. Hey, Michelle. All right, I see four people are watching. I know one of them is me, so I know I'm here. I know Lily's here. I know Michelle's here. And that might be the three of us. Nope, there should be one more. There should be one more. Who else is here? Someone else should be here. All right. I'm just gonna wait because I see more people are logging on. Just let me know that you're here. Say hi on the sidebar so I know you're here. And while we're waiting, I'm just gonna ask you in your notes wherever you're taking notes on this today. Just make a little Venn diagram. I want you to compare and contrast what you read in the book about the Ebola virus to kind of what's going on today with COVID 19. Definitely some similarities in the middle, but distinct differences on either side. And then I kind of want to put this in perspective to you in terms of evolutionary fitness, what this means. And we're gonna have a great discussion today. So, quick sidebar. Hey Sam, hey Dylan. Okay, take a minute in your notebooks for me right now. I don't know, like a minute, 30 seconds. Just compare and contrast, and then I'm gonna have you start, putting things in the chat bar that I'm gonna be typing in, that I'm gonna be pulling from you, and things I need to add in here and hopefully prompt you. So take about 30 seconds. I want you to start comparing and contrasting the two, and then we're going to start to really formalize this in our discussion. So take a minute, start comparing and contrasting. Yeah, she just sent me a text. Hi, Kyla. Kyla, my suggestion to you would be is I don't know if you're all using a BMCHSD account, but if you are and you're not able to comment on the sidebar, it might be a BMCHSD thing. So you may want to try logging in with a different email that's not school associated because I know people are clearly typing in. I don't know if it's a limitation because of your Belmore Merrick account, but try using a different account. And it still wouldn't for some reason. Kyla, is that new? I feel like I'm talking to you and texting you at the same time. Kyla, we'll figure that out because it might be a setting on your computer and it might be the device that you're using. So, let's start off with some similarities. Okay, what are some similarities between Ebola and COVID? Okay, start throwing down with me on the sidebar. I'm watching while you're typing, and I'm going to start throwing things in here that you are saying that I think need to get into this little area. I probably should type really, really small. And I'm going to make this a little bit bigger. Okay, start throwing down with things on the sidebar for me. Let's make you wider. Okay. Yeah. Okay, similarities. Give me some similarities here. Excellent. Okay, Lillian says that they were both considered zoonotic. So, Lily, explain to me what zoonotic means. Okay, that's Lily's task. She's going to start typing on the sidebar what it actually means to be zoonotic. And then my job today is to explain how the heck did we know they were actually zoonotic, because that's an interesting concept. Lillian also says RNA viruses. They both are RNA viruses. Anything else? Anything else that's similar here? Okay, Lily is going to give us an excellent definition of what it means to be zoonotic. And I'm waiting for someone else to throw down with some more similarities. We may have to come back to that, because that might be a little bit tricky for you, but it could be actually just really simple things, like what makes a virus? Like, why are they viruses? We can come back to that similarity in a minute if I'm not getting anything else from you, but you have about another 10 seconds to start typing stuff up. Okay, Lillian says it arises from another species and jumps to humans. Excellent. So, we had a species jump. Okay. Alright, let's start talking about distinctions. We may have to come back to that similarities in a few minutes, but let's talk about clear distinctions. Like, what clearly defines Ebola versus COVID? So, talk to me about that. Alright, so I'm going to come back here because Lily added something else. Both are capable of human transmission. Okay. Alright, I'm waiting for some more people to start talking. Okay, so Ebola definitely has a way higher mortality rate. Okay, so Ebola. Dylan, I'm going to ask you to, I don't know if you have the book in front of you or if you can remember, but there were a couple of different, classifications or relatives of Ebola. There was the Marburg strain, there was Ebola Sudan, and then there was Ebola Zaire, and then we also saw Ebola Reston in the book. Dylan, your task right now is to find the mortality rates for these guys. Find them. It doesn't have to be an exact number, but give me some quotes. Give me like, strongest or the highest, the lowest. Talk to me about that. Dylan, that's your job. I'm waiting for a couple other people to throw down with some more differences, similarities. Okay, Dylan, Ebola is a mortality rate. Ebola is a filovirus. Okay, so let's go over here and let's just talk overall structure. You're telling me, Lily, that Ebola is a filovirus or a filovirus? Which means it had like a worm slash like a whip-like structure, as opposed to, let's kind of compare this to COVID. What does COVID look like? Like, how do we classify those? No, not what I meant. little spoiler alert as to what's coming in my review in a few minutes. So let's go over here and let's kind of compare this, what we have going on with COVID. Why was COVID so different? And Dylan, you've been tasked with finding the Ebola mortality. Somebody with citation and source, just to make sure you're not, making this up. I want somebody to find for me the mortality rate for COVID-19 at this time. And keep in mind that this is a very, very dynamic case and situation because we don't really know enough yet. I feel like Marburg broke and then was resolved. Like Sudan, Zaire, and Reston, all of those outbreaks have broken and resolved. So we were able to get substantial data. Where the current mortality rate for COVID-19 is still very much so a work in progress. We're not entirely sure how this is going to play out, but what is the current rate that we're looking at right now? Okay, Sophia says the symptoms of Ebola are definitely more severe. So let's go over here. Sophia, I'm going to ask you currently to throw down with what some of the symptoms were that we saw in Ebola versus what we are currently seeing with COVID. Well, what are these symptoms? Like, what is there any overlap in symptoms that we saw? It's actually a really good thing to maybe compare the two because I think the symptoms can definitely be, distinguishable towards the end, but would you necessarily be able to tell them apart in the beginning? So, what were some of the similarities in the symptoms that we saw initially? Lily just threw down with something really excellent. Ebola's RNA is negative sense. Excellent. Let's go over here. I'm going to have to end up explaining this. So the RNA is negative sense in an Ebola virus, whereas when we talk about COVID, that is a positive sense RNA virus, and I did not intend to get into crazy detail with this today. This is not letting me copy. Because what I do plan on doing with you is one of the things I really would like to review with you is DNA replication. And the whole process of DNA replication. You really do not want to behave. I want to go over DNA replication with you at another time in a little bit more detail. And I think that might be the next stream, which will be the time when I get into what it means to be positive sense versus negative sense and what the implication is in terms of viral, viral stability, because that is definitely one of the things that they look into. Okay, so Dylan said the Reston fatality rate was 50. Okay, so let's actually go back a second because the Reston case was the one where the monkeys were in the monkey house. Did you guys read about the monkey house? I can't remember where I told you to stop. I don't know if monkey house is actually part 2. That's actually really interesting. I have to stop myself because I finished the book a couple days ago when I started reading. I love reading these things. And I don't think Ebola Reston came into the story yet. This had to do with the monkey house in Atlanta. Somebody just correct me, stop me. If you did not read up into the monkey house, because I'm having a little bit of a brain fart at the moment. I don't think you know about Reston. I think you know about Marburg, and the mortality of Marburg was 50% for humans. Sudan was different. Zaire, I believe, was something like 99, 100% fatality. Okay. Alright, so Lillian, thank you. The monkey house is part 2. So you actually don't know about the Reston strain yet. I'm going to delete that because that's actually an interesting story too. There's actually another strain of Ebola that we are aware of that actually broke on human soil. Not human soil, broke on American soil, but that's the next part of the book. All right, so symptoms. Sophia's throwing down with massive organ failure. I'm going to throw out there that you can definitely have some organ failure with COVID-19. It's not like full organ failure, but there's definitely some organs that begin to shut down. We're talking about massive, widespread organ failure. We're talking about massive hemorrhagic bleeding. We're talking about like the skin is separating from the body tissue. You are basically turning into soup from the insides. This is a hemorrhagic fever or a hemorrhagic virus. This is so not spelled right. Thank God we have spell check tonight. Hemographic. Hemorrhagic. Whatever. Okay, now this is really interesting because this whole entire little story started off with the guy got sick, and what was the first symptom that he had? I'm looking over there at what Sam's saying. Well, it starts off with a fever. It starts off with a cough. So if that's really interesting to me, fever and cough. Well, fever is a very, very uniform first primary immune response, so it doesn't surprise me that they start off with similar symptoms. Starts off with a cough, turns into body aches. I feel like these are all common symptoms between Ebola and COVID. Then you start talking about this divergence of these symptoms, which probably has to do with some sort of the viral proteins that are present, which is what I'm going to talk to you in a little bit with the symptoms and how we distinguish them. There's a couple other things that I just want to point out here that I was kind of hoping maybe someone would pick up on. Lily picked up on sense versus positive sense, negative sense. I'm really interested in the size of the viral genome. So somebody do me a solid right now. Go Google for me the size of the viral genome for Ebola, because that's an interesting thing we should probably talk about. And how many genes are in the Ebola genome. And I want somebody else to go find for me the number of genes that are currently, that we know about in the COVID genome, COVID-19, and then the size of that viral genome. Because that's something interesting that's going to play a role into where I'm taking you today and what I plan on discussing with you, but also that year was 90. Okay, so it also will play an interesting role into where I'm going with this story I want to tell you today and review. So Sudan was about 71%, Ebola Zaire was 90. Did anybody find for me a current mortality rate? Okay, so Sophia said the mortality rate for COVID-19 could be up to 20% in Wuhan, China, and that was where it initially broke out. So I'm going to throw over here. COVID mortality, they're believing was initially 20% in Wuhan, China. However, I believe right now, according to the CDC, let's just kind of hop on a, let's go to coronavirus. I want it's on my phone, hold on. Okay, 30 seconds. I want the coronavirus. I want the world meter. Coronavirus world meter. Okay, so at this current time, we're looking at the death rate to be about 16 with closed cases. But that doesn't take into account the number that are currently active. This is just a closed case. So how many people were resolved, how many people died, but there's so many more people right now that this number of 16% is really not accurate yet. We won't be able to know that until we start closing more cases, and we're like, we're in the middle of this. We can definitely get these values over here because these epidemics have broken, but then they also have resolved, and we haven't really seen a continuous widespread spread transmission that we're able to get further data. A lot of similarities here, a lot of differences. Okay, my first question to you is, in terms of evolutionary fitness, which one of these is fitter? In terms of evolutionary fitness, and you're a virus, which one of these is a better evolutionary strategy? I want to debate that for a second with you. Okay. And Sam just got me, not Sam. Yeah, Sam just got me some information I would have put on here. That's going to play a role in my little story today. This is about 18 to 19 kilobyte, which is 18,000 to 19,000 base pairs. And over here. And okay, Sophia, I'm gonna get into positive versus negative sense, but I don't think that tonight's the night for that. Only because it's gonna be the next stream probably. I'm looking for COVID's number of base pairs. And as soon as I have that, I'm ready to move on. Um, Number of base pairs. I don't see that on my chat bar yet. I think when I looked this up, it was 23,000 base pairs, but it might be 32. I might be having a little bit of a dyslexic moment. Hold on. I think it was 23. Should have wrote that down. Okay, I'm going to wait for one of you to find that, and then we're going to come back to that discussion in a minute. Okay, Lillian's throwing down here that COVID has a better viral strategy. It doesn't kill the host. Hey, Max, what are you thinking? Why are you going with COVID as being a better evolutionary strategy? Okay. While I'm waiting for an answer to that, I'm going to ask you another question, and I want you to think about this not just in terms of scientists, like the scientific population, but maybe by yourself, like you are an AP Bio, like you are have a scientific mind. What are some questions that scientists have about corona? What are some questions that scientists need to answer about corona in order to, help slow or stop the spread of this pandemic? What are some questions that you think scientists right now are trying to answer about coronavirus? Start throwing down on the sidebar with some questions that you think scientists are probably running through their heads right now that need to be answered. Okay, Max, you have a beautiful statement here. COVID definitely has a better evolutionary strategy because it does less damage to the host, which allows it to spread more. The host can interact more with other individuals, and it has a better chance at sustaining itself, which is going to lead into a discussion today because I feel like people think Ebola virus is so dangerous. And don't get me wrong, like I definitely don't want to catch Ebola. Like it's a death sentence. It is a very, very savage virus in terms of the symptoms. But in terms of evolutionary strategy, it usually kills the host. And it doesn't have a chance to jump from individuals to individuals easily. The viral load of Ebola is extremely high, which is like its evolutionary trade off. It is highly infectious, but it's not necessarily going to stay around for long periods of time because the hosts are dying. They're not able to spread. They don't have widespread community transmission usually in Ebola outbreaks like this that we're seeing with corona. It's like corona is silent. It's stealthy. Like it doesn't really let itself be noticed until later on. The symptoms become more severe over time. And then usually it doesn't, really manifest in all people universally the same way. They're not all, getting hemorrhagic fever. The symptoms are varied. Some people are coming in with fever. Some people are coming in with no fever. Some people are coming in with what looks like red, pink eye, and other people are coming in with GI symptoms. Other people are coming in with a dry cough versus a wet cough. People are affected differently because of that. It's a great evolutionary strategy because it's difficult to diagnose early and you're extremely transmissible during that early diagnosis period where you're not necessarily showing symptoms. So, okay, I'm looking right now at some questions. Excellent. Michelle wants to know, can people become reinfected? So that's actually something really interesting because that's going to change a really, really big strategy to solve this problem. Can people be reinfected? Yeah, that's definitely a big thing. Michelle, why would people become reinfected be a problem? Like, scientifically, not just like, oh, more people are going to get sick, but there's a really, really big problem with this if people can get reinfected. Okay, I'm looking right now at the sidebar. Does it inhibit the immune system? So maybe it has become more transmissible because it's slowing the immune system from stopping it. as an evolutionary strategy so that it stays in the host longer. The immune system's a little bit suppressed, so it's easier to spread. Okay, I'm looking for some more questions. Lily says, does temperature affect transmission? Lily, why do you think that this might be important? Okay, I'm still waiting for some other questions because I feel like you have questions that are great questions, but maybe start from the beginning. Like, I want you to pretend for a second you are on the front line. You are in Wuhan, China. Like, you see that there is now an emerging novel virus that ain't nobody's ever seen before. What are some questions that scientists had to have been asking themselves at that time? And then where did we go from there? Like, yeah, that's interesting. Those are problems we need to solve initially, but what are some other questions that scientists probably had? I'm waiting for a couple more questions before I go on because I'm going to throw down with some questions, but I'm waiting for you because I have a feeling that if I give you another minute or two, you're going to ask the questions that scientists were probably asking, at least in the beginning of this outbreak. Excellent. Does temperature affect transmission? So, could this become seasonal? Could it become a seasonal problem? Could this be something that we see a downswing in summertime, but a reemergence in the fall, just like flu? Excellent, Sophia. Sophia wants to know mutations? Like, what about mutations? Sophia, talk to me about your questions about mutations that could become worse. Okay, so mutations that could make this worse. So how about we just add in here, is this mutating? That's a really big one. And I'm happy because that's where I want to go today. That's part of where I want to go today. I'm waiting for a couple other questions. I want you to pretend one more time you are on the front line. You were there in the beginning in Wuhan, China. What are some questions that you have as a scientist? Excellent. Sophia wants to know, where did this come from? Where did COVID-19 originate? These are two massive questions. So my job today is, how can a scientist possibly answer these two questions? Because there's massive amounts of research going on right now. Like, I feel like the entire world, scientific community and medicine and pathology and, epidemiologists and the CDC and like the World Health Organization, these are massive questions they're trying to answer right now, and they're doing it. How are they doing this? What do they need to know in order to answer these two questions? And I'm not saying that this is not going to be important, because I can probably tie this in somehow, maybe today, if not the next stream. I might even be able to have to talk about this with you today, which is going to be interesting. And it's really going to depend on what's happening. Can it infect different cell types or only respiratory cells? Max Michael wants to know, how is it cured? And does it infect different cell types or just respiratory cells? I don't know if I can get into this today, although I might be able to briefly talk about this. Not necessarily I have a cure, but I can kind of give you some ideas as to where they're going. How does it infect different does it infect different cell types or just respiratory cells? I might even be able to address part of that today as well. So these are some big questions. The next thing I need to ask you is, if you want to answer these questions, there is one place you need to start, and there's one piece of information that you need to know about this coronavirus in order to talk about mutations, in order to talk about the origin or the divergence or the initial strain that COVID-19, like, where did it come from, and then its ability to infect other cells. Like, that is, I'm looking for one really important piece of information that you're going to need to know in order to solve this problem. Think like a scientist. You want to answer those questions. What do you need to know before you go any further? Randy, they're absolutely looking into malaria treatments like antivirals. Do I necessarily think that that is specific to corona at this time? No, I think what they're doing is they're trying every single antiviral that they have already to see if it is going to do any sort of, like any sort of mitigation to slow symptoms, to stop people from being so contagious. Is it necessarily curing? Is it necessarily targeted to COVID specifically? It's almost like taking Motrin, which you're not supposed to do. Apparently, Motrin's not good for this. Apparently people who take Motrin have increased symptoms, which is really interesting to me, and there's a whole biochemistry reason behind that I want to talk to you about, but tonight's not the night. I haven't read enough about it. But it's kind of like taking Tylenol when you have a cold. You could also use it if you have strep throat. You could use it if you have a migraine. You could use it if you I don't know, like you cut yourself or you broke a bone, like to take away pain. Is it necessarily treating, or is it just kind of making the symptoms, is it not actually curing? It's just making the symptoms less pronounceable. Excellent. Excellent, excellent. So Sophia says, I want to know about similar viruses. And Lillian says, the genome and the structure. So Sophia, I'm going to throw down here, because that's kind of a question. Where did COVID originate? And then, is COVID related to something we know in science? And in order to answer all of those questions, Sophia, you're on the right track here. I think Lily hit the nail on the head. In order for you to answer any of these questions, you need to know about the genome. And you need to know about the viral structure. And if there's anything hopefully you have learned in this class, is that, this whole class is designed around common ancestry and structure and form and function. Anything that we are talking about structure when we talk about viruses is some sort of protein-based structure. And any sort of protein-based structure is going to have some sort of genetic code, whether it's in the form of RNA or DNA. It has to have some sort of genetic code. So that's where I need to take you today. That took me about a half an hour to lead us into that story, but it's a great story. What I want to talk to you today is, how the heck do scientists answer these questions? And they need to get at the genome, and they need to be able to study the structural proteins that are present. So my next question to you is, well, how do we get at the genome? So now will be the time where you're going to take some notes, because one of the few things that I did not really necessarily talk to you specifically about, I feel like I kind of weave it in here and there through all different case studies, but there is definitely a couple pieces of biotechnology that I want to share with you today that scientists all over the world are using nonstop in order to study corona. I want to talk to you about, how do we study the genome and how do we look at molecular structures. That's my story today. So the first thing a scientist is going to have to do, if they want to answer any of these questions, when they want to get at the genome, step number one is something called PCR. So you're going to start writing with me and taking notes. This whole entire slide and, PowerPoint is going to be obviously available to you at any time. So don't feel, overwhelmed to start copying every single thing down that, all the pictures, because they're always going to be available to you. But PCR stands for something called polymerase chain reaction. And the way PCR, polymerase chain reaction, works is, I think of this like a molecular photocopier. I want you to pretend for a second you are a doctor and somebody walks into your clinic and they are showing signs of, some sort of novel virus that nobody's ever seen before. You want to get your hands on the genome. Well, where are you most likely going to find the viral genome? You're probably going to find it in the person's nose, because that's, like all the mucous membranes. You're probably going to find it in the back of the throat, the oral swab, and sometimes even in the cheek. Or you could even find it in the blood if it's some sort of bloodborne virus or some sort of bloodborne pathogen. So the first thing they're going to want to do is they're going to want to extract whatever, sample it is that they're going to be gathering. They take a swab of your nose, they take a swab of your mouth, they might take a blood sample. Now, you need to keep in mind that when you swab somebody's nose, you swab someone's, back of their throat, their cheek, you take a blood sample, you're probably not getting enough DNA in order to study it in high enough quantities. You're going to get some sample. You're not going to get a huge amount of it. And that's a little bit problematic. Like, you're going to need large volumes of that material in order to study it. So what scientists do is they do something called PCR, which is something called, like, it's called polymerase chain reaction, but I think of it like a molecular photocopier. There's a whole bunch of different pictures here, and I'm going to explain them all to you. There's 1, 2, 3 pictures. This is the PCR machine. And what a scientist is going to do, step number 1, is they are going to take the piece of template DNA. So I'm going to start highlighting. I'm going to kind of mix these two pictures together because they're both great, but they kind of do better jobs at highlighting different things. So I'm going to number my picture as I go, and I'm going to highlight. So step number 1 is they're going to take a piece of template DNA. This piece of template DNA, if you're talking about COVID-19, you're talking about some sort of, pathogen that you want to, study the viral genome that you're isolating from a patient, you're going to take the nasal swab, you're going to extract whatever, material's coming in, and there's a way for extraction. I'm not going to get into DNA extraction with you, or RNA, whatever material it is. We're going to take some piece of DNA, or this could be even RNA. We are going to extract the template piece of DNA. So this is step number 1, extraction. We have a piece of template DNA that we've extracted. Now, in order for us to do this molecular photocopying, I need to make another copy. So what happens is we have this process called denaturation. So step number 1, we have our template. And step number 2, we are going to denature this piece of DNA. Now, it's really, really important that we don't damage the DNA. We don't want to damage it, because if I start to break the backbone, then I'm going to have fragments. I don't want fragments. I want whole pieces of DNA. So I don't want to completely denature this. I just want to unzip the DNA. So step number 2, start typing. Step number 2, I am going to unzip the DNA. And this is done by gentle heating. Relatively gentle. So gentle heating, enough to unzip the DNA, but not enough to actually break the backbone. So what's really important here is that I want to make a copy of this DNA. So I open it up, I unzip the backbone, and then I need to actually make copies of DNA. And this is DNA replication in its beautiful, beautiful form. In order for me to make a new piece of DNA, I need to lay primer. There's primer. So over here, I'm going to actually box this up for a second. That's a lie. I want a rectangle. Over here, this is my starting materials. I am going to take this template piece of DNA, which if I highlight, the template piece of DNA is this dark blue. This is not highlighting. 30 seconds for my highlighter fire up. So I'm going to take whatever piece of template DNA that I have extracted from this patient. This is taking forever. Okay, this would be my template DNA. And then the next thing in this little picture is, well, I'm going to need primer, because in order for me to build my new strand of DNA, I need these DNA primers. This is taking forever. These are the DNA primers that are in picture number 2. So perfect, highlight. This is the template. We're going to call this primer. So we're going to highlight these in green, which is the primer. And then once I lay the primer down, and the primer needs to be, In order to make this work, I have to have primers that are going to be laid so that I can extend. I'm also going to have to add in some nucleotides. So these are nucleotides. And it's going to be a mix of nucleotides that I can then turn around and I can use to extend and build. So actually, this area over here that I'm highlighting in pink is going to be built from all of these little pink materials. I want you to stop and pause for a minute. And I want you to just pause with me and just make sure you understand the concept of when a scientist does PCR, they're going to take the template, and the first thing they're going to do in this little tube is they're going to spike the tube with nucleotides and primers that can be used in order to build new pieces of DNA. Take a minute, pause, give me the thumbs up to make sure we're on the same page. Okay, just give me some thumbs up to make sure we're in a good spot. If you're not wrapping your mind around it, now would be a time for you to tell me you're not understanding. Yep, I haven't gotten there yet. I'm going to add that, Lily. Okay, so in order for you to build DNA, one of the things that we learned earlier in the year is that during DNA replication, you need to have a DNA polymerase. Now, the problem is that in this machine, it's called a PCR thermocycler, you need to heat up this DNA in order to unzip it. But the problem is that when you heat this up, it needs to be heated to about 60, 70 degrees Celsius, and normal human body temperature is about, like, I don't know, like 36, 37 degrees Celsius. Like that's a heat that's almost twice as hot as, the normal human body temperature. It's going to be hot. Not hot enough for you to break the backbone, but hot enough for you to split the DNA. So you need a very, very special DNA polymerase in order to make this happen. And there is one other piece of material that's not in this picture, and that's the reason why I bought both of these pictures in. We also need something called, it's a special DNA polymerase. And this DNA polymerase is going to be, let me shrink you down. Not what I meant to do, but that's fine. Okay, this DNA polymerase is going to be a very, very specific DNA polymerase that is capable of working at ridiculously hot temperatures like 60, 70 degrees Celsius, and it is called Taq polymerase. And the reason why it is Taq polymerase is because it comes from a very, very special bacteria that was found in thermal hot springs that is able to tolerate extremely high, not extremely high, but really relatively high temperatures compared to human body temperatures. Otherwise, normal DNA polymerase would be denatured at such a high temperature in this machine. So we also have to put in DNA polymerase, and very specifically this thing called Taq polymerase. So I'm going to bring back my highlighter for a minute. And let's go teal on this. We're going to need Taq polymerase. I'm just going to put some Taq polymerases in here. And Taq polymerase's job is to extend these lines of DNA. So take a minute. Yeah, Sophia, 100% right. This machine is basically forcing DNA replication. Lillian said archaea time. Do you mean like it is like an archaea bacteria? It's definitely an extremophile. So it would be a bacteria that is capable of, living in thermal vents, and it needs to replicate its DNA. But in order to survive in a thermal vent, you better have an enzyme that is capable of handling extremely high temperatures. Yeah, Sophia, this is definitely DNA replication. But here's the thing: when this goes on, and I don't know, did I get one more picture of this? Nope. Okay. So the next thing I want to show you really quick, I'm just going to go back to this for a minute, because when I had Googled this, I thought I had grabbed one other picture, but I didn't. I want a picture of PCR. I don't want the thermocycler. I just want to show you one other image. And it's like this. I think I want this one. Okay, so here's what this would look like after cycle number 1. I would split it open, and I would end up with 2 pieces of DNA. Can I open this better? I just might need to snip you. No. No, 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 no. Sorry, there's a lot of no's that are coming out of my mouth right now. Okay, so I'm not going to be able to move my mouse. But in cycle number 1, I went from 1 strand, and when I'm done with cycle number 1, now I have 2 strands. Well, then what happens is that this is a thermal cycler, so once the second strands are made, then it cycles itself, and it happens again. So then once I make my 2 strands that come together, I'm going to heat it up again, split it up, denature it, run the cycle again. Now I have 4 strands. After I complete cycle number 2, awesome. I'm going to repeat the cycle. I'm going to heat it right back up, split them open, and I'm going to perform another cycle. So 4 strands just became 8 strands. 8 strands become 16 strands. 16 strands become 32 strands. You can do the math on it. It's pretty much exponential growth here, and usually a thermal cycle or PCR event, you usually run a thermal cycler for at least 25 to 35 cycles. So, that was like, 3-4 cycles. You went from 1 copy of DNA to 32 copies. You get the idea. By the time you're running, 25, 30, 35, 40, sometimes 50 or 60 cycles of PCR, you can end up with 1 piece of DNA that turned into millions of pieces of DNA that you are now able to study. We call this process amplification. And we amplify DNA in order to get a big enough sample in order for you to actually visualize it in a well. When you do gel electrophoresis, you need to be able to have enough DNA to get separate, stuck in the little weaves of the gel that you can actually visualize to the naked eye. You got to understand how many pieces of DNA actually need to be in that well for you to see something with the naked eye in terms of what's on a piece of gel electrophoresis. You need a lot of DNA. When you're talking about a nasal swab, or you're talking about, a blood sample, or you're talking about a throat swab, cheek swab, it's really not enough DNA. And the first step is always amplify. We need to do a PCR chain reaction, and we use this molecular photocopier basically in order to make 1 strand turn into millions of strands in this machine called a thermal cycler. It repeats, 25, 35, 50 times, and this is part of the reason why in the beginning, when they're doing samples and they're trying to determine whether or not someone had corona, this is the reason why it was taking them 5 or 6 days in order for them to actually get results that they could actually work with, because we don't have rapid technology right now where you could just do a swab and it's an antibody test and be like, yep, that's corona, or like, no, that's not corona. One of the things that the CDC was doing is they were turning around and they had to amplify the DNA in order to study it and sequence it and come up with ways that they could actually determine if this is corona and then find a quick test that can be done. So I want to stop and pause really quick and just look at the sidebar really quick. And Sophia says, how do they isolate the specific nucleotides? Do you mean, Sophia, over here? If you're talking about over here, they're not isolating specific nucleotides. Like, they're going into the chemistry stockroom and they are pulling out a sample of A's, and they're going to dump chemicals that, are adenosine into the test tube. Not going to dump because it's really small. They're going to micropipette, 15 microliters of A. And then they're going to turn around, they're going to pipette, 15 microliters of T. And they're going to have a mixed concoction with all of these nucleotides that they think they're going to need, and they put a whole lot of nucleotides in there because they're going to be building a lot of DNA. Okay, so Lillian says, is there any way to guarantee the DNA RNA is the one that researchers are looking for? So is it possible that, they swab and they do this test and they're swabbing the back of your throat and it turns out you don't have corona, you have influenza A? Absolutely. So what they would do is they would gather whatever piece of DNA or amplify whatever it is, and then they need to test it. They need to figure out, well, what the heck is this? And they were going to need to sequence it. I've only just brushed the surface of what happens when they're trying to identify a genome. So PCR step number 1, make a whole bunch of copies because we're going to need them to figure out the sequence, which is step number 2, something called Sanger sequencing. So I'm going to stop and pause, and I want you to tell me if you wrapped your mind around this, because in order for you to wrap your mind around Sanger sequencing, you had to understand a little bit about this PCR. So take a minute and give me the thumbs up. Okay. So PCR is the copying process. Now, if you know for a fact that you have whatever sample it is that you're looking for, that's awesome. But most of the time, as scientists, they're amplifying and they want to know, they want to know the sequence of what they're amplifying. They're not entirely sure. So sometimes what they will do is not only will they do PCR, but they will also do Sanger sequencing at the same time as PCR. And they'll run it at the same time. Slash, they will do this Sanger sequencing. It doesn't necessarily have to be at the same time, but the concept is they're going to denature the DNA the same way that they denatured it in PCR. They're going to split it up. They're going to heat it up and they're going to denature it. They're going to make multiple copies of the part that they're interested in, which is exactly the PCR component. They're going to run this a few times and make some copies. Now, the next thing they're going to do is they're going to attach a primer. Just like we did over here, we attach a primer because we want to build off of the sequences that we have. We're going to add 4 different polymerase solutions. So that's actually really interesting. I think there was another picture that I wanted, which did not show up here. Hold on one second. I thought I grabbed the picture that I was looking for. Wait one second. I liked this picture because it was kind of like a cartoon. Okay, they are going to be adding 4 specific solutions into the mix. Do you remember during PCR, they put the nucleotides in? They put in the nucleotides that we needed in order to build this. These are regular nucleotides. And regular nucleotides, when they are being built, regular nucleotides can be strung together along the phosphate backbone. They're able to be covalently bonded. These are regular nucleotides. Lie. Lie, lie, lie. I don't want a highlighter. These guys are regular nucleotides. Hold on. This is, the highlighter's just taking a minute. These are regular nucleotides, the DNTP. So they can be strung together. Now, not only do they put regular nucleotides in, they put these things called di-deoxynucleotides. And they have lost the ability, hold on, these are normal because they have an OH group on them. They have a normal OH group on them, which allows them to dehydration synthesis and continue the chain. These are the mutant ones. Right here. they're going to mix some of these mutant forms, which they're not able to extend the chain. So when one of these guys gets attached randomly, because you're putting in a mixture of normal versus mutated ones, they're not able to continue to build and the chain stops. What they then did was they spiked these nucleotides. If it is a C nucleotide, it's going to glow blue with a blue fluorescent tag. So when this stops, it's going to fluoresce blue. And there's other nucleotides, like there are T's, that are going to be tagged with a red I don't have a red highlighter, a red fluorescent protein, so that if this stops and it's a T, it's going to glow red. Or if it's an A, an adenosine, they're going to tag it with a green fluorescent protein, so when it stops, you're going to get a green glowy glowy tag. Or they're going to leave with G, there's no tag on it, so it doesn't really glow anything, but it stops because it is a mutant form that they have manipulated this OH group, so it cannot extend the chain. They're going to run the thermal cycler many, 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 many times. And if you have a random mixing of normal versus mutant, you're going to build chains normally, but if you pick up a mutant, you stop. And where you stop, you're going to get a fluorescent colored tag that tells you the letter at which you stopped. We're going to run this cycler many, 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 many times, and we're going to get copies made over and over and over again, and we're going to end up with a catalog where, like in this cycle over here, it ran for 15 bases and it stopped at a G and there was no fluorescent. So when it doesn't fluoresce, we know that it stopped at a G. And then we ran it again. Like we have another cycle that went. And in that cycle, when this stopped, it ran for 16 letters. And the letter it stopped at is going to fluoresce blue. And we correlate blue to C. We have an entire catalog that we will then run through a machine that is able to read how many base letters long this is and say it didn't fluoresce at the end. Well, that's 16 bases and no fluorescent, that's a G. And then they're like, oh, that's 17 bases and it fluoresces with a blue, that means that base number 16 is a cytidine. And then they'll be like, oh, number 19. Well, 19 ran until it was 19, and at the end we have a green fluorescent, and green fluorescent is going to match up with an adenosine. This process of Sanger sequencing is related to PCR in the sense that we're running multiple rounds and we're going to be cycling through this, but the cycles are not going to create identical copies like we saw with PCR. Like the whole job of PCR is to amplify, where in Sanger sequencing we're forcing this to stop, and whenever we force it to stop, we're tagging that endpoint with a fluorescence that we can then run through a gel. Here it is. And that gel can then be read through a machine, which will give us a sequence of what we call a chromatogram that is going to then turn around and tell us these are the DNA. This is the sequence of the not amino acids. This is the sequence of the nucleic acids that are making up this piece of genetic information. I want you to stop and pause and give me the thumbs up. Now would be the time for you to ask me if you're not wrapping your mind around how Sanger sequencing is done. Okay. How can you tell how many base pairs have passed? Lily, that's a great question. There are a couple ways to handle that. There are extremely very high resolution gels that exist. The gels that you all ran at Stony Brook, they were made with agarose. There's other types of gels called polyacrylamide, and there's other gels that, for the sake of me right now, like I can't remember them, but there are gels that have a resolution of like 1 or 2 base pairs, and you would be able to, like look at that. But now we actually have this all automated, that a computer system can actually scan through this information from a gel and turn around and has the resolution to distinguish between one base pair versus 6 base pairs versus 7 base pairs and pick up on the fluorescence. I actually want to see if I can find a picture of a Sanger sequencer. Hold on. And apparently I'm losing my voice. Sanger sequencing machine. Yeah, Lily, look how massive these things are. Like they're not like little pieces of equipment that like hang out on your desk. And I'm just curious, Sanger sequencing machine price. I'm curious how much these are. Oh. Some of these, the starter pack is about $1,000, but you can get the high end one, which could approach $1 million, and you can pretty much, it's yeah, that's crazy, insane. So yeah, these are not like little pieces of equipment. These are heavy duty pieces of equipment. Okay, so the next question. You now have a sequence. I want to go back a minute at your list of questions, and don't worry, we're not going to get into all this. I'm just going to show you some cool pictures. Um, so things that you asked. Well, does it related to something we know? Where did it come from? Could mutations possibly make this thing worse? Is this thing mutated? We got the genome, we got the structure. Now the next question is, well, I know the sequence. How do I figure out if this is related to something? What do I need to then be able to do with this sequence in order to determine its relationship to other organisms? Like, take a minute. And while we're going through the story, I just amplified, now I have a sequence. What do I do next? Lily, you're 100% right. Okay, Lily says we should blast. So, Lily, I was kind of looking for just a more common sense answer. I was hoping people would say we need to compare it, which is where we need to go next. We need to compare them, and the tool that a scientist would use is something called BLAST technology or the BLAST database. So, this is the snapshot that I took of the BLAST website. So if you were to go to, I'm gonna pull up another tab over here. If you were to type into Google BLAST, just BLAST, the first link you're going to get is from the National Institute of Health. And this is pretty much where everybody goes whenever they have some sort of piece of genetic information that they sequence and they want to know, is it something that I already know? Like, could that swab have been somebody just has the flu and it's, like something we already know about? That's possible. We would need to be able to compare it. We're gonna blast it. And if we blast this thing and it's completely like new and we've never seen it before, then there's not going to be, an exact match in the blast database for the genetic code, or there might be some similarities that we would be interested in. Since I have blasted last, since I've been to this page and I haven't been here, in the past couple, like months since I started, teaching AP Bio, I usually do blast towards the end of the year. This is brand new. So this whole beta coronavirus database, this is new in the past couple of weeks. This says it was established on Monday, February 3rd. That's kind of when the new cases were coming out in Wuhan, China. We're going to blast the sequence that we have. So what I would do is I would open up the database and I could put in my sequence that I have, or I would name my sequence and it would be uploaded. And I can then compare my sequence to an entire database that people have uploaded their sequences to. There is an area of this website, and I'm not, for this life of me right now, able to find it. You can upload your sequences, and then other people can start to blast their sequences against your sequences from all over the world to see, well, is this flu virus? Is it Ebola? Is it Corona? Like, what are we talking about? And we're able to compare it. So I want to go back to this for a minute. No, I don't. Okay, I want to go to this website and I want to actually open up the beta corona database. It's not what I want to do. I want to go to the bank. And if I were to start taking a look here, these are all sequences that people have uploaded recently. Like this was a sample from December 2019 in China. And here's another sample from the Philippines and Japan. And like, oh, these are like the Wuhan, China, sequences that they started to isolate from people. And the further down I go, we're going to see it when I start to compare, there's actually very specific sequences in Wuhan, China from people who were actually exposed in the market itself. So I can click on this, and this is someone's sequence that they have completed. And if I scroll down, this is the entire Sanger sequencing for that particular piece of information that they had extracted. Like that came from someone's swab that they then turn around and they Sanger sequence. And I think somebody said in the beginning that this was roughly, however many base pairs long. It's a lot of base pairs for you to sequence. So the next thing they would do here is this is a really tedious piece of information. So what they would do is they package it together. They give it a code. So this is the code for this piece of information. This particular sample came from the seafood market. It came from someone that was infected directly from the market. And I can go back to the home page of BLAST. I copied that sequence in, like the name of that file. And if I hit enter, if I hit BLAST, it's going to take a few minutes. Not even, it's probably like about 30 seconds worth of time. Right now, it is currently searching or blasting. Oh, it's actually really quick. It's blasting that piece of information to see what it is most similar to. So if I were to take a look at this, there's a whole bunch of samples that are 100% similar, 99% similar. These all appear to be the same version of the coronavirus, and this is interesting. This particular sample was from one of the United States cruise ships. And like these are individuals that they had sampled from the cruise ship. There's more Wuhan, China samples, cruise samples. What I find really interesting is that the further I scroll down here, it's getting less and less genetically related. It's starting to drop. And I did this earlier. I must have picked a different one. Hold on one second. Oh, wait, no, I just closed it. This is the one that I blasted initially. Because I think what one of the people, one of the questions that somebody had asked was, well, how do I know where this came from? So let me just go back to BLAST for a second. This was the sample that I initially picked because I found something really interesting here. So they're blasting this against the entire database. And the further I scroll down, it's not here anymore. I need to locate it because the one that I initially searched, no, I gotta locate it. The one that I initially searched, when this whole entire database came up and I started looking at it, it was really interesting because one of the last entries down here said 90% related to a coronavirus found in bats. So I think that this goes to answer this question. Where did COVID originate? Where did it come from? We're able to blast the sequences that we're getting from humans and we're testing it to see, well, what is this most like? And we're seeing that, oh, it's related to the same outbreak that was happening in Wuhan, China. Oh, it's the same, the same viral outbreak that was happening on the cruise ship. The ones that were isolated off the coast of California. Well, not only are people going to be blasting samples that they're getting from human beings as these viral outbreaks are going on. I'm pretty sure that in the documentary we watched with the different outbreaks, like the different pandemic videos on Netflix, we saw the part of the series where there are people whose job is to go to these markets and sample the different organisms, on a routine basis to see, well, what kind of things are popping up. And whatever they collect, they catalog and blast so that they're like, oh, we know what bat coronaviruses look like. And when they blasted this, and I really want to find that image, and I'm going to have to text it to you because I closed the one window that I thought I screenshotted it in. And when I was playing with my tabs, I closed the window. It was like an 89 or 90% match to the coronavirus that they found in bats. So they're believing that this has an origin that was probably some sort of jump that either happened directly from a bat to a human, or there was some sort of intermediate host where it passed from bat to something else in that market, but it was similar enough to bat in order to be pinged on the blast database. People are saying, well, how do they know what's coming from bats? Like, it could have come from anything in that market. You're right, it could have. But right now, it is closely resembling sequences that we currently have in blast for bat coronaviruses. That's where they're thinking that's coming from. So the last thing I want to leave you with here is, could mutations make this worse? And is this thing mutating? I want to go back to this. Because if you have the initial sample from Wuhan, China, and you're seeing that right now it's 99%, it's 99.9. The further I go down, I'm starting to see that there's some divergences. These percentages are kind of dropping over time. Like, some of these are really similar, but we're starting to see a trend of downward relatingness. There's an organization out there called the Next Strain, nextstrain.org. And what I find extremely interesting about this, if you get a chance, this is kind of like their philosophy, and I screenshotted this from their website, but basically their philosophy is they are going to be blasting and Sanger sequencing samples that are coming in from people all over the world. Like, the CDC is, reporting positive cases, but it's not like, oh, yes or no, positive or negative. They're turning around and they're sequencing it. And they're wanting to take a look at, is this changing over the course of the pandemic? This is Ebola. Hold on. That's flu. And this is what we're seeing. This is a map right now that is showing transmissions. And the initial outbreak started out with this purple area, which is a little bit colored over at the moment. But this purple area, they sequenced the initial strains that they were finding in the Wuhan market. And they're seeing that, well, over time, it was staying purple, but as it started to jump to different places, and it landed in Australia, or it landed in the Philippines, or it landed in, in France or Italy, it landed in America. It's almost what we call a little mini founder effect, where now you have this little isolated virus population that is then going to mutate in that area and have, little pockets of genetic diversity because of how it's jumping from place to place. And it's like, it's like geographic isolation, kind of. You're having little founder populations of this virus sample, and it's a way for you to trace, well, what parts of the world were infected with the Wuhan strain first. Did other areas spread the coronavirus that were not necessarily from Wuhan, China? Like, did they all start from China? And, there was a founder effect that went to America at the same time it went to Italy, at the same time it went to Spain and France and Australia? Or did somebody who went to Italy then turn around, that person traveled to South America? Or did somebody who went to Australia, did they hit up the United States? Like, what was the transmission pattern? And they're able to look at the sequences, and this is what they have found so far. The purple is the initial strain, the Wuhan, China strain, and as you start to see over time, we have some pocketing, and we definitely have variations in the COVID sequence. There are areas, this red area is the United States, if we take a look. The United States is the red area, red and orange, like the darker orange. The red and orange cases are in pockets of the United States. It's not to say that other areas have not had that strain or that not strain, but that genetic variation version of corona yet. I'm going to refrain from using the word strain at this time because I don't think that they are far enough divergent in evolutionary history to be distinct strains. They're more just genetic variants at this point. It doesn't mean that other areas of the world can't have the predominant United States of America sequence. We probably had a little founder effect where someone from the United States went to an area that was predominantly like this orangey. Hold on. So, like over here, whether it came from Europe to America or America to Europe, I'm not entirely sure I can say that from this diagram, but we're starting to see that there are variations that are present. So, this entire process of being able to track this divergence and this change that we're seeing through this organization called Next Strain, that's this guy, we can only do that and determine, could these mutate, or is this thing mutating, if we actually know the sequence and we know the structure, which is kind of what I took you through today, the PCR, the synchro sequencing, the blasting. That's awesome, but really what this leads to is, I want to know, how are these things related. This is the Ebola outbreak, so the different strains of Ebola. There's Ebola Zaire, Ebola Sudan, Reston, there was the Marburg case. This is a whole little hot pocket way of them being able to trace, viral outbreaks and look at it in terms of the genetic similarities. This is what goes on with the flu. I find this diagram extremely interesting because if I look at the United States of America during the flu seasons of 2014 to 2020, I don't know if I necessarily see any founder effects here, which to me, a handful. To me, this means that this is more of a what's the word right now? I totally just had a brain fart. Endemic, which means that it becomes almost natural in the world of humans. It becomes part of our everyday viral outbreaks that exist. This is not novel because we do have such a widespread variation of all these strains in the population across the world, as opposed to what we're seeing here. We definitely have some speciation events going, not speciation, but we have some genetic variation that over time could definitely lead to new strains, which is my final thought to you. Even though this thing is mutating, there's a whole bunch of really cool diagrams that you should check out from nextstrain.org. These are all the same different ways of representing the same information. I just think that's really cool. The last thing I want to leave you with is all of these mutations, like, they're happening. Do they signify vast changes in phenotype? Do they necessarily make them more dangerous? Like, does this necessarily mean that they're going to be more infectious? Which I think is this question that Sophia asked. Is it mutating? Could mutations make this thing worse? And clear as day, this thing is mutating. Like, it's part of biology. It's constantly mutating. It's an RNA virus. But does it necessarily signify vast changes? And the answer to that question right now is maybe. It could, but not necessarily. If I start to change the DNA code, there is redundancy. So it doesn't necessarily mean that if I change and I have a change in genetic information, like I'm changing the DNA sequence, I'm not necessarily changing the amino acids. I can have multiple DNA sequences that are coding for glycine. I can have multiple DNA sequences that are coding for threonine and valine. But it doesn't necessarily mean that if I change the DNA, I change the amino acids. And even if I do change the amino acids, I could be changing one hydrophobic for another hydrophobic. It doesn't necessarily mean I am changing the physical phenotype. It doesn't necessarily mean that. But it doesn't necessarily mean that these mutations can become drastically different. And in order for us to actually have that conversation, we would need to actually take a look at the protein product, which is a whole nother stream for a whole nother day. I'm going to take you through the protein database, which is what I was playing with right before we logged on together. I was playing on the protein database, and one of the new things that just came out, I know we played with this earlier in the year, but there's an entire section right now on coronaviruses and all of the proteins that they're taking a look at, because even though the DNA sequence or the RNA sequence changes, it's really about the protein product and finding out, does the protein product really change? Do the proteins on the structure of the virus really change? Could the protein of those structures turn around and allow it to infect different cell types? Because right now we are seeing that it's infecting primarily respiratory cells, but you start to take a look, and we're starting to see more and more younger people are becoming more susceptible. We're seeing younger people die. Maybe it was just that we didn't have enough data when this thing first came out, or maybe this thing is changing. And we have to look at, even though the DNA sequence is changing, we don't necessarily know how it's impacting the protein product. And then the last thing would be here, well, if it's impacting protein product, can people be reinfected? That definitely could change that. Like if I change it enough and it's definitely a different protein product, then my antibodies are not going to work. Um, how is it cured? That's going to be a whole discussion for protein product, which I'm going to take you through the protein database, protein folding, protein sequencing, relate it back to Corona for you, and I think that'll be our next Corona Chronicles discussion. All right, I think I'm done. I am looking right now at the chat bar, so just give me the thumbs up and make sure we're in a good spot, and then I think we're calling it a night. Okay, I'm gonna close the stream. I'm gonna be updating the questions again for part 2 of Ebola, and I'm going to find a way to tie more concepts into review. As of April 1st, I believe, the AP College Board is going to start giving me a little bit more guidance as to review videos that you can be watching through the College Board that I will start posting review assignments with. And there's also things in the College Board, student portion where I can assign practice problems to you for review. So we're going to start heading into that too. I'm going to have you read part 2 of the Hot Zone. We stream again most likely on Monday. We get through some more concepts of review, and then early next week, I'm going to start putting together a game plan using College Board AP Central so we can start hitting the concepts that we're going to see on this modified AP exam. So have a wonderful night. Happy reading. Happy couch surfing. And I will see you all soon. Have a wonderful night. I miss you.